Hello, everyone. Welcome to the review of EULAR 2023. I have with me the faculty who covered lupus at EULAR 2023, and this is our lupus panel, where we gather and share notes about what we thought were the highlights of the meeting on systemic lupus erythematosus. We're going to begin with introductions. I'm Jack Cush in Dallas. Janet?
Janet Pope, London, Canada.
And Bella?
Bella Mehta, New York.
And Yuz?
Yuz Yusof, Leeds, United Kingdom.
Excellent. Okay, so let's begin. Why don't we begin with Bella?
Hi. So I think there was many interesting abstracts, including the guidelines. But let's talk about one of the abstracts that I particularly like. It's poster number 0170. This was looking at all-cause mortality and cancer incidence from a large registry. So this was like around 1,500 patients, around 6,000 person years, and they followed these patients over time to see incident cancer rates and also compared it in patients who were on biologics versus those who are on what they call standard of care, but what I assume it's or in the fine text, it's mostly DMARDs. And it did notice that there was slightly higher incidence of cancers, like the standardized incidence ratio was like around 1.21. But when they adjusted it, incidence ratios, for example, for rituximab or belimumab, those were mainly the two biologics that they looked at, was not, the cancer rates were not higher. Again, this is sort of reassuring data in terms of biologic use in SLE patients, but also that, overall the risk was not very, very high in lupus. I know that cancer risks and even mortality risk, there's a lot of talk in the RA world, but not in the lupus world. So this was a good reassurance sort of a study, given the low rates of mortality as well as incidence cancer.
So Yuz, my recollection of this information is that there are some studies that show higher cancer rates, which we've always assumed would be related to inflammation. But lupus patients, it's not as high as an RA because the patients are younger, and we're not using cytotoxic therapies anymore. Where does this stand in the lupus community?
Yeah, so I do agree. So I think this is a beautiful study that presented by Bella. I think we need this data to reassure our patients because I do face that in the clinic. A lot of our patients sometimes, when I wanted to start them on immunosuppressive therapy, such as mycophenolate, because it's written sometimes in the information leaflet that it can increase risk of cancer, and a lot of patients sometimes they come back to me saying like, "Oh, well, I've read this information leaflet about it, and there's potential increases of cancer, but it's not quite specific to mention what potential risk is there." So I think this data is quite reassuring in that sense. Yeah.
Okay. Go ahead, Janet. What's your first one?
Okay, so this was a large RCT. So looking at Upadacitinib as monotherapy in SLE, and just a couple things. So it's a large phase 2, so they're going to go into phase 3. So it was a combo molecule that was a BTK type drug with UPA 30, another molecule with UPA 15 of this BTK, the molecule alone of the BTK, UPA alone at 30, and placebo. So four arms that were active treatment of 2 different doses, and then the placebo. So lots of people probably were feeling that they had active drug because it was a high chance of having active drug. Anyway, good primary endpoint, SRI-4 improvement, steroids less than or equal to 10 milligrams, and that was at 24 weeks. And what they wanted to look at was SRI-4, but the overall flare rate, what was happening with double-stranded DNA, and then a little bit about the interferon gene signature scores. So number one, The added molecule, the BTK, didn't do anything. Number two, we're talking about 30 milligrams of upadacitinib. And if you remember that UPA is a higher dose potentially in inflammatory bowel disease, ulcerative colitis and Crohn's, and they will have, if they haven't already in many countries, gotten their approvals there. It's also a higher dose in atopic dermatitis. So this higher dose here was well tolerated. There was SRI-4 was no added value with the two drugs combo instead of the one drug. So BTK didn't do anything. And the placebo rate was kind of modest to moderate, which we do expect. No new safety signals, including no MIs or no imbalance of VTE or PE, DVT when you look at that. So I thought it was interesting. I think, after a failure, which might have been trial design, bad luck, who knows, of a JAK2, knowing here that a JAK1 is successful and knowing that in a way that competition for an oral treatment would be from the Paisley trial, the Baricitinib, which is the TYK2 drug. So I think it's kind of interesting, maybe a new potential, another oral treatment, and we'll see what both these drugs do in phase 3.
I thought this was encouraging. I think it was interesting that they threw in the BTK inhibitor in there and that they had a combination of a BTK inhibitor plus a JAK inhibitor. But you're right, the BTK did nothing, and hence that's the end of that story. Although in the United States, we had one BTK inhibitor approved for hematologic reasons this year, but there are trials in a lot of autoimmune disease, MS, lupus. I heard scleroderma, certainly RA, but I think this is one of the first ones where we're going to see it's not going to be developed any further in lupus. I'm sure there are other trials going on, so that's the end of that. But will the JAK inhibitor last? This is a phase 2, and it seems to me, when I keep thinking about why lupus drugs fail in lupus, especially when they look like they're going to work in phase 2, they usually fail because placebo responses. I want to blame the outcome measures, or the drug isn't good enough, or the patients were wrong. But boy, high placebo responses are what killed ustekinumab and baricitinib. And when the placebo responses stay low, so if I'm designing a trial myself, I'm not going after an SRI-4, I'm going after a renal endpoint, but I'm doing all I can to keep that placebo response low. What do you think about these placebo responses, Janice? And I mean, you're a real methodologist.
Right. So what I think you have to really do is disqualify when you go up on your prednisone more than transiently. Then you should be an intent to treat analysis. If you have to up your steroids for more than 2 weeks or something like that, and certainly not near an endpoint, that's a primary or a secondary endpoint, then you're out. So you can still keep them in the trial, but they'd be an intent to treat, that they would be considered non-responders. Once you start letting the prednisone kind of be fiddled around, where one group is actually lowering on average more, and the other group, the placebo, is increasing on average more, to me, that shows that one drug works better than the standard of care. And I think a lot of trials haven't done that. And I think that really contaminates studies too. And this one, I think they did adjust for, they looked at the cumulative steroid dose. But I think you just, RA trials were never done where you could mess around with prednisone for very long.
No, you're out if that happened. And, a lot of the RA trials, I mean, it makes sense that lupus, we include standard of care. So we did it in RA, but RA, the standard of care is kind of uniform. It's always methotrexate and very little else. Whereas in lupus, the standard of care can be hydroxychloroquine, azathioprine, methotrexate, mycophenolate, and those are not like running the same race, treating the same disease. And so that also kind of, I think, makes doing lupus trials really difficult and may contribute to some high placebo response rates. But all right, let's, I think that was encouraging though. We look forward to further studies with upadacitinib. Yuz, what was high on your list from the meeting?
Yeah, so I just want to discuss regarding to therapy again. So I think this about Baricitinib phase 3 trial in lupus nephritis. So the reason I want to bring it out is because of once the session finished, I think everyone's just muttering around in the room saying like whether is this going to work or not for lupus nephritis. So I think it's causing more questions rather than an answer. So as we know, a trial of baricitinib in non-renal SLE in 2 phase 3 randomized controlled trials, BRAVE1, which actually had a positive signal, whereas the BRAVE2 actually have a negative outcomes. So therefore, the company said, well, I think the trial results is inconclusive. So what happened next? So now we're looking in terms of the lupus, so with the renal population. So this is randomized, well, the investigators claim as a phase 3 randomized controlled trials in Egypt, so in one country only. So what happened, they randomized 60 patients into 2 groups. One is baricitinib, and second, plus placebo infusion monthly, and then the other group 2 is cyclophosphamide. So it's quite a good dose, monthly cyclophosphamide plus placebo tablets. So the primary endpoint that they use is reduction in proteinuria. So they only just use one single measure rather than more composite endpoints. So what they found, the primary endpoint was actually at 12 weeks, so it's quite short duration. So at 12 weeks, there are 70% people on the baricitinib group achieve a significant reduction in proteinuria versus 43%. And also when they did adjust for like SLEDA and complements, also at 24 weeks as well, it shows that it's significant in the baricitinib group. So I think what's the thing that we're not so people are debating about it, because in terms of the endpoint measure used, so there's only single like just proteinuria, usually these days, like in terms of complete renal response rate. So we have like lower proteinuria plus stabilization of creatinine or eGFR, and also reduction of steroid. I think that will be ideal. So if that will be used, other thing to mention as well is I think people are a bit nervous because what happened is quite brave, the study. I mean, it's fun, from the previous trial, because they're comparing. It's not like baricitinib plus MMF versus, treatments like that. It's just baricitinib versus cyclo. I think people are a bit nervous whether is this really going to work on this high-risk population. So usually on lupus nephritis, we're actually comparing to the standard of care. So I think that's the thing. I think the more question to answers, I think we need a larger studies if this is actually been developed in this indication further.
So they get accolades for doing such a study at a single center with 60 patients with nephritis. That's really quite impressive. Yeah, we'd like to see larger numbers and whatnot. But even though they chose a single variable outcome being proteinuria, which is not a great monitoring drug nor a great primary endpoint, they had the same pattern of outcomes with secondary endpoints as well. So there were a number of measures that were acceptably better in that baricitinib group. So that's encouraging.
The population,
what's that?
The population that they're doing this, right? It's very homogeneous there, but I don't know if it'll be reflected when it's like, African Americans more. I don't know, like with Caucasians. We'll see what that throws in the mix.
So they're homogeneous because they're genetically and ethnically homogeneous, as opposed to in the U.S. where it's Hispanics, whites, African Americans, Asians, and that makes
the response rates for lupus nephritis are different in these patients, right? In all these groups, we know that.
Yeah.
The other thing is no one's going to develop it because obviously Lilly has walked away from the drug in lupus after failing their phase 3. But I think it gives us a chance that maybe if patients, our standard of care in North America and actually in UK would be MMF, not cyclo. But I realize in a lot of countries, cyclophosphamide is less expensive and not an inappropriate standard of care. It's just CellSup would be our standard of care. But I think it gives us, if my patients have not responded to a few things, and maybe I would consider this because it's very difficult for us to get access to double dose belimumab, which is what you need for lupus nephritis because of the cost. We don't get voclosporin because of the cost, and anifrolumab we don't have yet publicly reimbursed in Canada. So, and they're working on a double dose as well for their phase 3 RCT and lupus nephritis. So I think right now it's like, okay, if we have to, it might be something we might consider.
You know, Yuz, I think Bella brought up what was the secret sauce in this trial, and that was the homogeneity, both in the population and the endpoint of lupus nephritis. You know, going with a general SRI-4, I don't know who to give the drug to in the end when you have that endpoint. And then lupus nephritis with you know, activity parameters going in. When you homogenize your population, you get a truer answer. Do you think that that's right? Does that, you know, how do you feel about, you know, a organ specific outcome as opposed to a general outcome in lupus trials?
Yeah, so I think that's why I think people wanted to do more clear in one organ specific, for example, that renal nephritis, because potentially you can get a clear picture of what's going on rather than the non-renal side where there's plenty of domains, you know, occurring. However, I think even these days, like to submit for regulatory approval, I think you've got to use as composite endpoint measures. And if you compare, we now have 2 drugs that actually, you know, licensed in lupus nephritis, and belimumab and voclosporin. So they've been using this composite index. So I think, you know, for the next, you know, drugs that's going to be marketed, so you need to have a unique selling point, you know, to outlast, you know, these tools now that have been licensed. So yeah.
Okay. I'm going to start where our next round of shorter presentations. Abstract 0586 was an abstract about something we have talked about at ACR last year and recently, and that's a better biomarker for disease, and that's CD163 in the urine of patients with lupus. This wasn't as big as Dr. Andrea Fava has done at Hopkins, but 45 patients with lupus of different types and whatnot. And what was shown quite simply is that it's a very discriminatory marker between active lupus and inactive lupus, between active nephritis and inactive nephritis, and performs better than many of the other things that we commonly use. And I think we need to be going, I like this research, although it's uncontrolled and not as large. Again, it supports the work that is starting to come out that says urinary biomarkers really have greater predictive value. We rely on what in managing lupus? Complement levels, not always great, and proteinuria, certainly not great. They have pitfalls. When they work, they're beautiful, right? The problem is that they're not truly predictive enough to have the utility that they're certainly given. So that was my one shorty favorite that I liked. Okay, Bella, why don't you give us yours?
I think the second one I would say is abstract number OP0226. This was, I think, a large, a 13-country large prospective study with lupus patients, looking at we really want remission in all our lupus patients, but even if you achieve lupus low disease activity state, seems like there is less organ damage, less adverse outcomes, even in a prospective follow-up arm that they had. They had around 4,000 patients looking at least 2 or 3 years of data. And I think it just sort of takes home the fact that not just remission, even low disease activity is probably a good target for these difficult patients sometimes, because just achieving that also improves our outcomes significantly in terms of flare, damage, mortality, and the numbers look pretty promising there. So, yeah.
So I saw this. I didn't get to see. I saw the title. I circled it. I didn't get to see it. Am I right in that the take home on this poster was that there are advantages to LLDAS over remission in the lupus cohort they looked at?
Not LLDAS over remission, but even LLDAS is probably protective in some sense It's not better than remission. Is that what you're asking?
Yeah.
No. I think that they were just saying that even just LLDAS itself is an independent predictor. It is even without remission. But like if you achieve LLDAS and then remission, obviously it's better.
Again, and again, it's
these
LLDAS,
guys-
right?
Right, LLDAS.
Yeah,
These
LLDAS.
guys published and presented a year or two ago that if you were in remission, you had better survival, and the longer you were in remission, obviously, the better the survival than sometimes. So it's interesting. It's not contradicting. It's just almost like a dose response. Remission's the best. LLDAS is pretty darn good. Higher disease activity, not great. So I think they're just cutting and slicing and dicing, but I actually like that one too.
Yeah.
Can I ask each of you, I'm sorry, can I ask each of you, what do you measure in your lupus patients to determine if they're well controlled or in remission? Do you do LLDAS or anything like that, Bella?
No, I don't.
I don't. Janet?
No, except for research cohort.
Right. And Yuz, what do you do clinically in your practice?
Yeah, so I don't use LLDAS clinically, but we do measure BILAG in our clinical practice, in routine practice. We've got a nice performer, easy BILAG to use as well. Yeah.
Okay. So the British Isles lupus activity, I don't know what the G stands for.
Yeah, group.
Group,
Group.
Yeah.
There you go.
I think.
I mean, I think our patients are already asked so many questions sometimes that they don't answer patient-reported outcome measures well if you don't dedicate a particular amount of time for it
Right.
on the visit.
Okay, that's interesting. Janet, okay, Yuz, go ahead.
Can I just add, in addition to Bella's posters, the work that presented, also Professor Eric Moran's also presented in oral presentation. I think it's OP0226. So I think also a large cohort in Asia, in Pacific, in terms of the use of LLDAS, and what they found that if you reach LLDAS, you will actually be protective in terms of flare and damage. So not, if you can stay there longer, it'll be quite good as well. But I think even if you just reach the LLDAS target, you still get the benefits of that. So I thought we're just adding on that. Yeah.
Okay, that's good. Excellent. All right, Janet, what's your last one?
Okay, so I'll be brief. I think a couple years ago, a year ago, I tweeted, "Don't put the cart before the horse." And now I think we should maybe put the cart before the tea and think of CAR-T. So I'm starting to really change my mind on it. It seems like it was well tolerated. Now, number one, it's not ready for prime time or every patient. Number two, in many groups, it'd be very expensive. And number three, there's only 7 patients. But these patients that had CAR-T, it was well tolerated. They weren't really getting a cytokine release syndrome the way they do with lymphoproliferative. The cells can be saved if you're recurring. They can be sort of unfrozen and given to you again. And these people come off therapy in order to get the CAR-T treatment. Obviously you got to drain their lymph cells, and the more you deplete them, the better, frankly. They get the CAR-T in there, they grow their B cells back, and they're naive. So they're not making DNA. The complements have normalized. Some antibodies stay, like the ones you'd like to stay from the plasma cells, which would be all your immune system of your memory cells from infections and vaccinations. So it seems to be the plasmablasts that are happening, and the B cells grow back naive. They're not really tickled yet and maybe never to be making antibodies and autoantibodies from lupus. So it's only 7, the largest follow-up presented was only a couple of years, but these people have remained in drug-free remission and in biochemical and antibody remission, which is pretty cool. So I think we need some actual randomized controlled trials, because if we can reset someone's immune system in a disease like that, where these people have failed a lot of drugs, and a lot of them had nephritis, like they weren't trivial disease. I think it's pretty neat.
So Janet, at the ACR presentation by Georg Schett, he showed that I think with 6 months of follow-up, I think only one patient was starting to get back their B cells. Now we have an extended follow-up now on 7 patients. What's the story on B cell return in these patients? Is it still staying down, or is it starting to come back, but they're still in remission?
Right. So what's happened is the B cells have grown back. After you do CAR T, your cells are supposed to proliferate. That's what happens at day 7. After you get it, your B cells are growing, but they're naive in that they're not recognizing the antigens. So they basically aren't producing, they aren't recognizing. So that's pretty cool. And by the way, it's it was OP0141 for those listeners who want to look. But anyway, so far so good on the B cells. One person had a bit of a relapse, was retreated. I don't know how you prime them the second time around. I'm not sure, but they were retreated because you make more than enough of the CAR T for that individual patient. So they were retreated and have done well.
Excellent. Okay, Yuz, you get to close out the session. Tell us what you liked.
Yep. So I'm a B cell guy, so I still hope in terms of B cell therapies. So the one that I want to present is phase 2 RCT. So this is Ianalumab. So this is a different mechanism. So it's a dual inhibition. So it's actually a BAFF receptor inhibitor, but it acts on 8 ADCC, which kills the B cells, and also it then acts on BAFF, which inhibits the B cell survival. So Ianalumab got a positive phase 2 trial in Sjogren, which was presented 2 years ago, and now in phase 3. So now it's following in lupus as well. So this was an interim analysis of the trial. And I think we talk about this now, but the placebo response is now, Jack. So essentially what happened in this trial, so it's a non-renal lupus. The primary endpoint is quite strict. So it's actually using the SRI-4, but also combined with prednisolone lower than 5 milligram per day. So the trial of Upadacitinib just now was 10 milligram per day. So so they compare Ianalumab versus placebo and plus standard of care. So what happened in the interim analysis, so there were like 34 patients in each group. So 44% achieved this primary endpoint at 6 months compared to 9% at 6 months in the placebo group. So it looks like the data is reassuring, the safety profile is reassuring as well, and it's now open to start phase 3 trial, and something that maybe we will look forward to in the next few years. Yeah. Mm-hmm.
So where was the trial done?
So the trial was done actually multi-center, a few countries. Yeah. Mm-hmm.
With a 9% placebo response. That's
interim analysis. Yeah.
That's really it's impressive, and that's a ticket to regulatory approval, if you ask me. So that was great. The low placebo response was again seen with the telitacicept, a repeat study that we have looked at before, coming out of China. That also looked good, different mechanism, but so I'll ask your final comments. I think most of you had some or were able to view the lupus guidelines. Does anybody want to give a one takeaway from the lupus guidelines for the audience? Janet, you want to start?
Sure. So one is prednisone's bad, use less of it. Try to get them to the lowest dose, 5 milligrams or less, and hopefully less. You don't need to pulse maybe with 1 gram times 3 in lupus nephritis, maybe for some people 500 milligrams times 3. For other sites, or even just starting like 40 milligrams orally and rapidly going down and adding other immune suppressants with it. So prednisone is bad, less is better.
Bella, do you have a takeaway from the guidelines?
I think the IV cytoxan, they put that back in thinking in high-risk renal failure patients, you can give IV cytoxan. And also the combination drugs, like now they're saying we can give combination stuff. I mean, we do it in clinical practice all the time, but now it's in the recommendations, give rituximab, Benlysta, and others as needed. So
excellent. And Yuz, what do you like?
Yeah, so I think the most progress is pertaining to the lupus nephritis, in addition to the low dose steroid that Janet was mentioning. So the recommendation is they said for remission induction, first line with cyclophosphamide using the Lupus or MMF, and then there's a line, and then considering combination therapies with voclosporin, belimumab. So I think. For me, I think I probably would like to be a bit more harder rather than considering, because of the data clearly shows, these days belimumab and voclosporin versus MMF, they're superior than MMF. So I think the practice these days, I don't think so we can really justify patients, we justify our act, like to give MMF first and then fail 3 to 6 months later, and then you add something. So I would like to see it more aggressive. So I'm actually co-chairing of the British Society of Rheumatology revised guideline, which is going to come out next year. So I don't know how my committee going to react with if we're trying to recommend it stronger, but we'll see from that point of view. But I think, but it's good recommendation that they acknowledge the combination therapy, but maybe I would just like to put a bit more stronger and considering. So I think that's probably my take. Yeah.
All right.
Some people had the other way around, right? Some people were like, oh, they're promoting these combination therapies much more than they should. So you have like a totally, I understand it works and you should do it in the right patient, but not in all patients too.
I know I'm thinking of the nephron that we may lose. So.
Well, the guidelines and the grade methodology favors the companies who do the studies. So if you do the study and it's going to be a grade A evidence per, in the review, you're going to be in the mix. But I must congratulate EULAR for also being pragmatic and practical about some of their statements here. So that, there is no new data on cyclophosphamide, but yet it still is in the mix, as is mycophenolate. So, and as you know, guidelines are really for you to consider. You're not bound by them in any way. They're good teaching tools, if anything, and good ways to have discussion. So, all right, I want to thank the panel for an enlightening discussion and for all their hard work at EULAR 2023. It was a great meeting, largely because of Janet, Yuz, and Bella. Have a good night, everyone.
Thank you. Bye-bye.